interesting because we have this unique team structure. So as you have all been talking to me, I have seen lots of great progress. You guys clearly understand this stuff. And the goal today is to solidify that understanding even more. Next. So again, as you guys are aware at this point, this paper was concerned with a machine learning problem about learning the structure of biomedical relationships. So it is agnostic to the specific kind of relationship, and this paper is really discussing a process. And the reason why this paper was so interesting to me, besides the fact that it was well written, is that it's very indicative of all the different factors that you need to take into account when you have an end-to-end data science pipeline. So you'll see a lot of this paper was talking about how do we process the data? How do we source it? What kinds of considerations and treatments do we need to think through before we even throw things into an algorithm? And that's really interesting to me because as practitioners of machine learning, you guys are going to want to internalize all of these ideas because this paper does a really accurate job of showing just how much work goes into constructing a solid pipeline world, and then tie that back into conclusions. What are the limitations? Where do we want to take this research in the future? As well as what did we not quite understand? What was ambiguous about the way they frame things? So I have a couple thoughts on all of these points, but again, it would be fantastic if you guys, as you have different considerations, different points you want to bring up, throw that in the chat, and then we'll just talk through some of these things if you're still confused. So with that being said, Quan, does your team want to go ahead and give us a little bit of a breakdown of the biomedical context of this problem?
And drug-drug interaction is important for understanding drug response and drug-drug interactions. And these papers are manually curated and then put into these knowledge bases like OMIM, Drug Bank, and GKB. And because of the copious amount of papers that we have, we need to find a way to automatically obtain and use all of these information. And I think that's why this is so great.
And I think just to build on that, right, the reason why I love the way you're framing this context is because you begin with the financial stakes, right? I mean, it's clearly a very expensive process to go about trying to manufacture, discover drugs, and any of the subsequent relationships. So it seems like these kinds of automated processes remove a little bit of the human oversight, but the benefits you get are saving millions of dollars or potentially years of human labor time, right, when you're trying to assemble this information. So it seems like even a single new proposed target can have an incredible financial payoff, right?
Yeah, because that's the part of the Stanford parser when in the paper they say that they're just using the raw Stanford parser without alternating anything so that to simplify everything for the reader.
Exactly.
I think that's a huge area to improve on because you can just take Stanford parser like code pipeline and then you change it.
Exactly. That's such a great point, right? They make this very clear. They say their main goal, right, is to keep things simple, and they focus on the process, right, not changing every artifact of what the process outputs. So we have an area to improve on this for exactly the same reasons you're saying. So thank you so much for that breakdown. I think that did a fantastic job of setting up the rest of this. So then we're going to talk about the dependency piece of this, right? How does language processing take this raw data that has been framed by Quan and his team and then translate that into an actual object that goes into the core algorithm here? So if we could get a brief summary and overview of the dependency parsing, that'd be fantastic.
And I'll introduce for the second slide. This is more than what we will use in this research, but I think knowing this will make a better understanding of dependency parsing. There is another kind of parsing called the constituents parsing of a syntactic parsing on previous slides, and the right slide is the example of the dependency parsing. It only shows the interrelationship between the object and the subjects. So it's between words. In our case, it's the drug-gene relationships.
So there's obviously going to be a distinguish emphasis on what each sentence means. So in order to take away any confusion and to make everything clear, these kind of rules need to be defined by very easy to understand terms so that no one else is confused. And that's kind of where dependency parsing can be very, very useful because certain terms must mean certain definitions. So there's very few, places where people can make error. And in this case, we would like to be able to display the relationships between words and specifically between drug and gene.
Dependency parse, and then do we have to combine the two things to pass into EBC? I think that was kind of our confusion, and we would like if we could get some clarification.
Yeah, definitely. Thank you for bringing that question up. So I can take an attempt at answering it, but does anybody else content? I think that your diagram pretty cleanly breaks this down. I don't know if you or somebody in your team wanted to try and answer that piece, which again is what actually happens going from the raw text to input to EBC. What are kind of the overall steps in between that happen there?
Yeah, I think I can answer this. Unless someone else want to answer before me. I feel like I've talked a lot.
It's never a bad thing, but where it's actually showing you what are the linkages between the two things you're interested in the sentence. So that D piece is one example of a column inside the matrix that Quan was just talking about. So like he said, every row is a potential pair of things to be connected. Every column is a binary indicator suggesting, are they connected by that particular dependency pair?
In the roles,
Mhm.
the drug gene, so it's every drug gene pair. So like there's gonna be a lot of roles.
Exactly.
Okay, yeah. I think that
Yeah.
was kind of the confusion.
And just to build on this idea, right, the reason why they're talking about sparsity, right, a matrix sparsity, is the fact that you need to look at every drug gene pair with every possible dependency path that occurs in your data set.
Yes.
So that's a big cross product, right? There's a lot of different cells there. So that's why they say it's very sparse. And that's why EBC is also uniquely poised to handle this problem. So I know Wafa's team is going to be taking on the actual algorithm itself. Dependency paths mean the same thing, even though they use slightly different words. And the way you find that out is what kinds of drug gene pairs are they typically connecting, right? So that's the overall idea of the modeling. And I want to leave the rest of the discussion to the remaining discussion for EBC. But hopefully that helps you to orient yourself on how that works operationally.
Yeah, that was very helpful. Thanks.
In language and leverage them more effectively. So again, this is how deep learning tends to work, is it's built off of progress that other teams have made. So you may have heard of, pre-training or pre-trained models, and dependency parsing is a model that has already been trained, right, on a specific set of data. So let's keep that in mind as we move forward, that the original Stanford parser had limitations in the way it can accommodate biomedical language. And so other parsers actually go out of their way to get that piece correct. Does that make sense?
Yeah, yes, it is. And
Great.
yeah, it brought up I remember reading it in the original paper, an interesting point. As you say, like this is a limitation, but I remember the authors addressing that because I think it operates on the level of like drug gene pairs, kind of it's more robust to like these errors as long as the errors are consistent.
Exactly. That's a great point, right? So they say this, as long as the system is misbehaving in a stable way, right, we can go ahead and estimate these relationships, affect things. But that's a great point. So hopefully that makes sense. And the dependency piece is becoming a little bit clearer. But you guys did a great job of breaking down the theory.
Matrix with and the rows are the drug gene pairs and the columns are the dependency paths. So the first step is the unsupervised by clustering, in which we co-cluster the rows and columns into clusters into a predefined number of row clusters and column clusters, in which the drug gene pairs that are connected with similar dependency paths are clustered together, and the dependency paths that connect similar drug gene pairs are clustered together. The co-occurrence matrix is an n by n matrix with n drug gene pairs and also paths data from the Medline data and arrange it in an n by n matrix. The second step is the unsupervised, which uses the ITCC algorithm, and it records the number of runs in which each row co-clusters with another row. The supervised steps, you identify a seed set and a test set, and then score the test set according to how many times it co-clusters with the seed set. So the ITCC, which is the information theoretical clustering algorithm, which is used in the unsupervised step, in the co-clustering step.
Awesome. Yeah, thank you so much. So just to talk about how this will work operationally, I think that there are two very clear bottlenecks, right, to accomplishing this replication. One is that the dependency parsing is difficult programmatically, right? It's that translation from raw text to the data matrix, right? And they say,
But if
you know,
anyone else
AUC,
has any thoughts about that, it would be great.
yeah, that would be fantastic if anybody has any input on this, because this is something that I think is easy to just take for granted, right? I mean, when in replicating this, it's easy to just compare things to 0.7, right? AUC is an easy metric to generate with any library, but we want to think a little bit, right? Whenever we see a statement like that, it's so easy to gloss over and just move on to the next piece and say, okay, that makes sense. You know, higher AUC is a good thing, right? We want high AUC. But when somebody just picks a threshold like that, you want to ask yourself why that have changed any of the results. And we don't know the answer to that yet. And that answer might be really important, or it might not have any impact at all. But these are the kinds of things when you're critically evaluating a piece of research, you always want to ask yourself, do I follow every step of the way here, right? So I think the EBC algorithm motivation is so well written, it's very easy to understand why they took that approach. But in evaluating it and scoring it, it's a little bit unclear to me exactly how they proceeded there. So I would just make a note.
A single sentence can contain multiple drug genes. So after dependency parsing, there are near about 2 lakh unique drug gene dependency paths in Medline, 4% of which connect at least 2 drug gene pairs, and 50,000 drug gene pairs co-occurring in Medline.
10 or 20 or minimum 30. Now, as you climb, right, the total number that you need to see, that sparsity is going to start to drop, because by the time you get to something like 100 or 200, let's say,
Yeah.
Cool.
Okay. So this EBC outperforms actually other classifiers on both the datasets. Like you can see, like EBC for dense dataset on PGX task gets an accuracy of 89.6, where a non-EBC gets 31.3. So there's a large difference. And for sparse datasets, it's even more. Like it is getting 54.4 on PGX task, and non-EBC getting like 1.1%. So there's a huge difference. Like
And
EBC outperforms.
I just have a quick question on that. So what are the non-EBC classifiers?
The non-EBC classifiers are like the average cosine and rank sum, and EBC is like EBC rank sum for calculating the AUC. Like you can see these graphs, like this is the EBC rank sum, and the deep blue is like average cosine, and the light blue is like rank sum. Okay, so EBC is getting 0.7 AUC, and other are not getting that much. Okay, so like EBC is outperforming other classifiers.
About a question,
Yeah, go ahead.
Colin, I have somewhere in the paper that they said EBC take into account the semantic similarity between two dependency parts also, while the other classifier didn't take that into account. So that's why it's create a huge difference in the evaluations.
That's a great point, right? So that's kind of the fundamental idea, I think, right? Is one method can actually leverage semantic similarity and one cannot. And we want to think very deeply about this, because all over the place in machine learning, you have an option, right, of how complex you want your model to be. Some models can leverage semantic relatedness via computation. Some models actually explicitly learn contextual word embeddings, et cetera, in an NLP context. Some models simply take, it's a good point.
Okay,
So I had a thought experiment for this.
Yeah.
Like here we were using ITCC, but like you mentioned, like there are some ways to, other than semantically getting the relationships between two tokens, use embeddings to kind of get a distance as well. Do you think that embeddings in this context, maybe like my intuition is that it won't work as well because it's just drug gene pairs and like you kind of need that semantic information and relationship that connects the two. But do you think that maybe like embeddings could be a area of extension here, or do you think they'll fall short in comparison?
Yeah, that's a fantastic point. So some of you may not know what a word embedding is. It's basically a vector representation, right, of that particular word or term. And the reason why that's an interesting approach is a great point to bring up, right, is once you've embedded a word into a vector space, you have some geometric properties associated with that word, right? And so by geometric properties, we mean if two words are very close in an embedding space, they typically mean the same thing. So synonyms if they're embedded properly, should be almost identical in embedding space. And then different dimensions of the embedding space can correspond to different concepts or differences, right, between two words. So I think that it's a really interesting idea, right, because we know it's possible. And Matthew, would you mind going back for 1 second? I just want to make this point. So I really love this breakdown, right? We have a visual, we have all of the interesting pieces of that visual to the right. So I think this is going to form a nice reference for groups trying to understand, again, the full final set of outputs. And I would highly encourage you, if any part of this visual is confusing, you want to kind of remedy that understanding and clarify. Important too.
Yep, I think that's a good point because I think this beautifully encapsulates all the different areas into just one visual, like you said. And it's very clear because these numbers are just the clusters. And at first, my group was I think we were kind of overwhelmed because there are many elements in this picture. And we were kind of diving headfirst into it. But as we read and understood the research paper more and more, we kind of understood the motivation for why they created this visual like this. And
most people.
Also, Colin, I think I have the answer to your AUC question.
Okay, let's definitely let's absolutely talk about that because that's really interesting to me. And the final thought before I have to hop off is, again, I really love the way we're building off of each group's presentation. So that's what this is about, right? Is each of your own perspectives is going to be useful for some component of this. So we want to string it all together